Recording? Mitosis is a process of cell division in eukaryotes that results in genetically identical daughter cells. Before mitosis occurs, the cell spends most of its time in interphase, which is the stage where the cell grows and prepares itself for cell division. In interphase, the nuclear envelope is present and intact. The two chromosomes, centrosomes, form when a single centrosome duplicates itself into two, each with their own central pairs. Centrosomes are regions in an animal cell where the mitotic spindle, made up of microtubules, is organized. Chromosomes are duplicated during the S phase. However, at this stage, the chromosomes are uncondensed and cannot be individually identified. During prophase, the chromatin begins to condense in this stage, and the duplicated pairs of the chromosomes are condensed to form two identical sister chromatids, joined at the centromere. The nucleolus disappears, and then the centrosomes move away from each other towards the ends of the cell. The centrosomes form small arrays of short microtubules called asters. Microtubules coming off of the centrosomes form the mitotic spindle. In prometaphase, the nuclear envelope becomes fragmented and the chromosomes continue to condense. Meanwhile, the microtubules extend from each centrosome to enter the nuclear area. Now, for each of the two chromatids, there's a kinetochore, a specialized protein that connects itself to the chromosome and the mitotic spindle to allow the kinetochore microtubules to pull the chromosomes around. The non-kinetochore microtubules go to the opposite poles and elongate the cell. Metaphase is the longest phase of mitosis and involves the chromosomes lining up single file at the center of the cell. The plane on which the chromosomes line up is known as the metaphase plate. The centrosomes have spindle fibers that attach to each sister chromatid with a protein called the kinetochore. The centrosomes on either side play a tug-of-war game with the duplicated chromosomes at the center, each tugging a sister chromatid to their side of the cell. Anaphase is the shortest stage of mitosis, often lasting only a few minutes. Anaphase begins when the cohesion proteins are cleaved by an enzyme called separase, allowing the two sister chromatids on each chromosome to separate, becoming their own chromosomes. The two daughter chromosomes begin moving towards opposite ends of the cell as their kinetochore microtubules shorten. Since these microtubules are attached at the centromere region, the chromosomes move centromere first, and the cell elongates as the non-kinetochore microtubules lengthen. By the end of anaphase, the two ends of the cells have equal and complete sets of single chromosomes. During telophase, the cell divides into two genetically identical daughter cells, each forming their own nuclear envelopes. Cytokinesis, or the process of the division of the cytoplasm, begins during telophase. In animal cells, a cleavage furrow develops, which consists of a contractile ring of actin microfilaments that is activated by the protein myosin, pinching the cell in two. In plant cells, a cell plate forms by vesicles bringing in materials to the middle of the parent cell. The cell plate eventually develops into a cell wall as the vesicles merge and the two daughter cells are formed.